Now students, in today's lecture or video, we will study amino acids with acidic side chains and amino acids with basic side chains. Now firstly, we will start with amino acids with acidic side chains. In previous lecture, we have already studied about aspartic acid, glutamic acid, aspartate and glutamate. Now, the amino acids aspartic acid and glutamic acid are the proton donors. So at physiological pH, these amino acids are fully ionized, that is, they contain a negatively charged carboxylate group. This COO minus is the carboxylate group. So the fully ionized forms are called the glutamate and the aspartate. So this figure is representing that this COO has negative charge and we're calling it as a glutamate. So after deprotonation of the glutamic acid, we will get glutamate. Now after deprotonated protonation, we will get aspartate. So after deprotonation of aspartic acid, after loss of the hydrogen, we will get aspartate, and in this case, this COO will get a negative charge. Now, what are the amino acids with basic side chains? So these are the amino acids which have ability to accept the protons. So at physiologic pH, the R group of the lysine and arginine are fully ionized and positively charged. So these are positively charged at physiologic pH. Now, in contrast, the free amino acid histidine. Histidine is a weak base and is largely uncharged at physiologic pH. So this histidine contains a R group which is either positively charged, protonated or neutral depending on the ionic condition which is provided to that protein. So this is the figure which is representing that we have histidine with this structure, lysine with this structure and arginine with this structure. We have to memorize these structures of amino acids. So the most important property of histidine is to contribute in the buffering system. So histidine is the only amino acid with a side chain that can be ionized in within the physiological range. So histidine, what is the structure of histidine? This histidine, this is a structure of histidine. Histidine contains an imidazole group. This is the imidazole group. So histidine is the only common amino acid which is in the ionizable form, which is can be ionized in a physiological pH. So in many enzyme catalyzation reaction, this histidine, which is abbreviated as HIS, facilitates the reaction by serving as proton donor or proton acceptor depending on the condition. So here we have discussed the amino acid with acidic side chain and the basic side chain. Now in our next video, we will discuss the abbreviations which are used for amino acids.